So most of the reactors I was saying in industry are working with configurations which have already been standardized with microbial fermentations. So what you will observe is the same reactors are modified for plant cell cultivations. So but one has to be clear how plant cell cultivations would differ from microbial fermentations. So in terms of size, microbial cells are pretty small, 2 to 10 micron size, while plant cell suspensions, the aggregate size, there can be cell clumps which can go as high as 50 to 100 cells together, and the size of the plant cell itself is much larger, which is around 20 to 40 microns. So you can imagine there is a big difference in the entire aggregate itself, the size, while microbial cell suspensions will be uniform. They are not self-immobilizing in nature. Then homogeneous suspension, that's what it is. So microbials are homogeneous, but in plant cell cultures aggregate formation would take place, because of which there will be eventually, with high cell density, you will see non-homogeneity arising in the reactors. Then growth rates in microbial fermentations, it is quite rapid. The doubling time is in hours, while in plant cell fermentations the doubling time is in days, around 2 to 5 days. So which also means that the reactor cultivation time would be much longer than the microbial fermentations. So keeping aseptic environment, maintaining it, the sterility maintenance also becomes one of the critical factors here. Shear stress tolerance, it is not that plant cells are highly shear sensitive. In comparison to microbial cells, they are less shear tolerant. The reason being because they have vacuoles, so the cell contents are stretched onto the cell membrane. Majorly this would be the scenario when you are working with matured cells, or if the production phase is happening with the matured cells. So there is one large vacuole, so the cytoplasm and the cell contents are stretched onto the cell membrane. So there is cell wall, so more rigid structure, so more brittle in nature. Then shear stress tolerance, so because there is presence of cellulose based cell wall which is of high tensile strength, makes them more sensitive to shear and less flexible. Aeration requirements, although specific oxygen demand of plant cells is much less than microbial cells, so what will happen, initially you would not like to have as much aeration as you would see in microbial fermentations. So because the specific oxygen demand is less, but as the cells would grow and there will be high cell density, aggregate formation will take place, so which will lead to what? Which will lead to mass transfer limitations. Now in order for the oxygen transport rate to increase such that the oxygen reaches till the inner core of the aggregates, OTR has to be manipulated. Now because oxygen transport rate has to be manipulated, the only controls in our hand are aeration rates, then increase in RPM. So this is what is generally done. But then balancing between the shear forces which will get generated over the oxygen transport rate which is required has to be done. Then so despite the fact, why it is important, despite the fact that specific oxygen demand is less, but the oxygen requirements for plant cell fermentations will keep on varying during the cultivation. So cultivation time with continuous fermentations in microbes, you will see it can go up to days, 2 to 10 days or so. But in plant cell fermentations, if you go to continuous cultivation, then it can reach up to a month or so. Product accumulation with microbial fermentations, it is generally extracellular, but with plant cell fermentations, you will find it is often intracellular. So which means what? The impact on productivity is much higher, because anything which impacts the cell with product forming inside can lead to change in the yield of the product. Then also if you want to make the process continuous, downstream is affected, because again there is an extra step of disrupting the cell wall and taking the product out. The cost is involved. Now what are the implications on bioreactor design with these factors? The differences between the two, the microbial fermentation and plant cell cultivations. Lower oxygen demand than microbial cultures due to low metabolic rate, that was one. Specific oxygen demand is low during exponential growth, but high oxygen transport rates are required at high biomass concentration. What does that mean? I just spoke about this. Specific oxygen demand is low during exponential growth. Specific oxygen demand is low. That's fine. What is specific oxygen demand? The amount of oxygen per unit time per unit biomass is low because the metabolism is slower in comparison to microbes. But what is high? High oxygen transport rates are required as the biomass concentration is increasing. What does that mean? The two are different. Specific oxygen demand, which determines what? The overall oxygen uptake rate. So, with increasing biomass concentration, the oxygen uptake rate is increasing. So, oxygen transport rate has to be increased, and moreover with cell aggregation happening. So, therefore, variable oxygen transfer rates are required during the entire cultivation period. More shear sensitive than microbial culture. So, this means what? That cultivation has to be done under low shear conditions. So, what can be done? You will generally observe that Ruston turbine is used for microbial fermentations, 6 plate. It is the most commonly found configuration of the impellers. But in plant cell fermentations, marine impellers or cetric impellers or centrifugal impellers, which can give better mass transport rates, because better KLA you will observe are preferred, and with less energy dissipated or with less shear forces generated are preferred over these high energy dissipating impellers like in microbial fermentations. Higher diameter to width ratio is preferred. Large diameter impellers are preferred. Why? Rather than small impellers with axial movements. Radial impellers. In microbial fermenters, radial impellers. Ruston turbine is a radial impeller. Now, in plant cell fermentations, you will observe that axial impellers are preferred, or cetric, which can provide both axial and radial. I said aggregation happens. If there is not much suspension created, the cells will settle down, which will eventually lead to loss in productivity in the critical phase when it had to happen, which was end of the log phase. So, then you need a good suspension ability, and that higher suspension is created. As I spoke about that the axial movement helps in suspension. So, and large diameter would create mixing and higher suspensions, axial impellers, and less shear generation. Now, cells often grow as aggregates or clumps, unlike microbial cultures. So, mass transfer limitations can limit the availability of nutrients to cells within the aggregates. Then cultivation has to be done under low shear conditions. Cell to cell contact. Now, we also have been now hearing that even though what one would like in a reactor is free suspension or well dispersed homogeneous suspension, but plant cells do not. If you try to create, this is what they are also observing. Now, while preparing the inoculum even or optimizing the shake flask, what initially people who have worked with microbes but they don't know the requirements of plant cells, you would try to keep your uniform suspension as uniform as possible. So, but then eventually you will observe that there is a loss in yield of the product, because we now know that cell to cell contact leads to a differentiation signal which helps in secondary metabolite yield and productions. So, there has to be a balance between homogeneity and even cell to cell contact. Plant cells are self-immobilizing. Sometimes when you are working with tissues, you will find that the growth would not begin unless all the tissues come together. What I used to observe during my research when I was working with hairy roots, when we used to inoculate roots, the roots used to be very small in size. We used to cut sections 2 centimeter longs and we used to then inoculate into the shake flask. Initial 1 week, it used to take, although we used to put the required amount of inoculum, but the culture or the growth was not happening. Why? What we used to observe is, until unless all the roots fragments which we had dispersed in the medium, the growth used to begin only when everything used to come together and they used to form a clump. And then we used to see that log phase used to begin. So, which is an indication that it depends on the species also, but self-immobilization is a prerequisite for the growth to begin. So, that is why sometimes you will observe that support structures are needed. Immobilization of your cell mass is needed. Maybe you need any immobilization like polyurethane foam inside. This is what we used. We used polyurethane foam then while designing reactors, where we could inoculate the roots so that they could stick together, and then the lag phase could be reduced. So then all that has to be optimized. Then cell to cell contact can influence secondary metabolism in plant cells. So optimal degree of aggregation self-immobilization is required for product synthesis. Then volatile compounds. Now because you are trying to, with high cell density in the reactor, what you would do? You would try to increase the airflow rate so that the oxygen transport rate can improve. But then you may observe that your secondary metabolite yield has gone down, because the volatile components like carbon dioxide or ethylene, which is crucial for your secondary metabolism, it plays a role in as a signaling compound, then which may affect the secondary metabolite yield. This is again what we observed with hairy roots. We then eventually optimize the inlet gas composition with carbon dioxide also. So plant cells, they are more sensitive to other gaseous components like carbon dioxide or ethylene, so which is needed to be optimized, unlike the microbial fermenters or fermentations. So what all things have to be kept in mind when you are designing reactors or reactor operating strategies for plant cell fermentations? The shear environment in the reactor configuration, the oxygen transfer capacity of the reactor, mixing mechanism in the reactor, foaming and aggregation. It is observed that in plant cell fermentations you will see much more frequent foaming than the others, because of the large amount of sucrose which is used, along with the proteinaceous, the growth hormones which you use, which might alter the surface tension, and because of which the air bubbles are not allowed to escape, so they stabilize the foam. So there is much more foaming in the plant cell fermenters. Then aggregation, which can eventually lead to mass transfer limitation, has to be taken into account. Maintenance of aseptic conditions for relatively longer durations. So as I said earlier, because the doubling time is in days, so your batch time generally varies from 2 to 3 weeks. And if you are planning to do continuous, it can go even beyond 1 month or 1.5 months. So that is the time period which we are looking for maintaining sterility. Then capital investment, obviously, that is common, be it any fermentation. That has to be looked into. You cannot end up in making such a complicated design which is not able to get scaled up, or which might be incurring a lot of cost. So modulation of factors like mixing and aeration is to balance out the mass transfer requirements and the shear sensitivity. So what are the factors which will affect the reactor design? Let's talk about the rheology. Now, rheological characteristics of plant cell cultures, what does it say? Plant cell cultures, they are viscous. So as the cell density increases, they tend to become non-Newtonian in nature. Non-Newtonian means? So which means your viscosity will also, now it is not no more proportional to viscous force or your shear forces and shear rate, but it becomes a function of, it is not constant, it becomes a function of shear rate or even density. Acts as non-Newtonian fluid, so shear stress and shear rate correlation, it depends on aggregate size and cell concentration and thereby fluid properties. Now culture rheological properties will get affected by medium viscosity and osmolarity. What is osmolarity? Now viscosity would keep on changing, so we can understand. What about osmolarity? What is osmolarity? It is also used as one of the indirect estimations, ways to indirectly estimate your biomass. So what is it related to? Now that you have guessed from osmo.
Salt concentration
Inside the cell. So solute concentration in the reactor. Osmolality per unit gram or moles of solute per unit amount of the solvent. Osmolarity is the amount of solute per unit volume of the solution. Now, cell aggregation, foaming and wall growth in plant cell fermentations. Now, plant cell suspension cultures, they form aggregates. So, diffusion limitation can happen within the aggregates. So, there can be external mass transfer limitation. So, when we are talking about oxygen transport rate, we are only taking into account what? The external mass transfer limitation, which is your KL or kg. And we neglect the KS part, the solid liquid mass transfer. We were neglecting it till the point. Why we were neglecting it? Your KL is what? Is the overall mass transfer coefficient, which involves only the gas liquid boundary. But we are neglecting the solid liquid boundary. There will be a solid liquid limitation, boundary limitation, isn't it? What solid? Because of the cell. So why we were neglecting?
Earlier the cells were very small.
Very nice. Because it was made for. So you could assume that. But if you are actually working with plant cells or tissue cultivations, it depends on the size and the size of the aggregates. If your cell suspension is uniform, you are much closer to that assumption, you can neglect. But if you are working with tissues much larger than the bubble size, then can you neglect that boundary? No. So internal, and moreover when you are working with aggregates or when you are working with immobilized cultures, can you neglect the internal mass transfer limitation? Internal means from the surface, solid surface till the inner core. People have even found that when they are working with root cultures, there are intracellular gradients of oxygen diffusion limitation. So oxygen gradients are observed till the inner because the size is appreciable. Now, foaming is frequently encountered in plant cell bioreactors with aeration. Initial, so I have already told you, because of the proteinaceous medium sometimes, or the proteins which come out once the cells are lysed because of the shear forces or the high sugar concentration. Now, wall growth due to cell flotation creates thick layer of necrotic cells. So, plant cells, as I said earlier during my discussions, that they have a tendency to adhere to the glass walls. So, you will observe in lab scale, especially lab scale reactors or non-movable parts. So, because of the large size. So, once they get stuck there or once they adhere to those walls, then subsequent growth will cause a film or an aggregate size to increase. As it increases, then the innermost layer will be devoid of the nutrients, availability of nutrients. So, then you will observe necrosis. So, mixing to keep the cells in suspension and to provide homogeneous environment. Now, at high cell densities, culture viscosity makes the mixing difficult because they tend to aggregate. Now, there has to be a balance between homogeneity and suspension. So, now both the things, if you try to make it very homogeneous, what kind of reactors would you or impellers would you prefer? Large shear forces and even for good oxygen transfer. So, you would choose impellers which can create more amount of shear force or more energy dissipated onto the medium, so that the bubbles can be well dispersed inside the medium, and there are more circulation velocities, radial velocities generated, so that it is homogeneous in nature. But then this will eventually lead to larger shear forces. So, you will observe that impeller design is a crucial thing in plant cell fermentations. Shear where what is to be considered? The amount of shear which is generated, power which is required, because that will involve cost. So, power dissipated, power required to drive the impeller, fluid circulation ability of the impeller, then under high viscosity, then large. So what is preferred is, you will observe in plant cell fermentations, large diameter impellers are preferred because of their high homogenization but less bubble dispersion. So again, although there is lot amount of, you can improve mixing because you have larger diameter impeller now, but what you are giving away is, because then this will create lesser shear force on the sparger for the air bubbles to be dispersed. So what will happen, eventually you will observe lesser dispersion of gas bubbles, lesser oxygen transport rate. So things have to be manipulated during the fermentation as the plant cells would grow. And baffles. Now baffles we know enhances mixing, but you will observe baffles in literature are found to be detrimental for plant cell cultivation. Shear forces could be reduced around the baffles. So people say that it is better to run plant cell fermentations without baffles, because mixing requirements or air dispersion or oxygen transfer requirements are much lesser than the microbial fermentations. So you can afford to remove baffles. Now in pneumatically agitated vessels, superficial gas velocity affects the suspension efficiency. Now there is no moving part. So in your airlift reactors and bubble column reactors, what is causing the circulation currents? The air bubbles. So particle suspension, and that is what is going to keep the cells in suspension, suspended state, not allow them to settle. Now particle suspension improves with high reactor aspect ratio. This is what has been observed. Higher is the aspect ratio, which is, aspect ratio means height to diameter ratio. Higher is the aspect ratio, better is the suspension ability. For the same power input per unit volume, so that is one of the power per unit volume is scale up criteria. So for the same, for power per unit volume keeping same, mixing time in airlift reactors has been found to be much longer than the stirred tank. Isn't that obvious? Now mixing is a limiting factor for pneumatically agitated reactors at high plant cell concentration. So even if you are choosing an airlift reactor for plant cell fermentations, because there are no moving parts and the air can keep it suspended, but then mixing is a limitation. So that has to be taken care as the cell density would increase. So which, with everything, don't you think I am giving you a pro and a con, a merit and a demerit? What does that mean? So how will you choose? Why am I giving you a merit and a demerit? Everything has a merit and a demerit. That's what I can understand from this. So what does this mean? What is needed? Right. So which means that depending on the species, don't think that at not knowing your culture requirement, you can just begin with, okay, now I have optimized everything in shake flask, let me do a batch now in stirred tank reactor. No, it never works like that. Your batch, once you have even optimized everything in shake flask, you need to take it to the reactor. Choosing the right kind of reactor configuration would be very crucial for reproducing the results which you have obtained in shake flask at the reactor level. And then once you have chosen the right reactor configuration, then the second stage of scale up begins. That what should be the right scale up criteria now to increase the volume of this reactor configuration. So oxygen demand and supply, dissolved oxygen levels to be maintained above the critical concentration. So that is same for any kind of fermentation. So we need to ensure that the dissolved oxygen, the bulk oxygen concentration in the reactor is always maintained above the critical oxygen levels. High culture viscosity, cell shear sensitivity and cell aggregation would make your oxygen transfer in plant cell fermentations a challenging problem. Now give me a way, just think logically. I know that cells would be shear sensitive, my cells are aggregating, assume they will be aggregating, and I can't increase the too much air and RPM because that will create shear. What would you do then? We have now learnt about so many things in optimization strategies. What would you do to improve your biomass or your yield with this limitation? Cell shear sensitivity, culture viscosity, cell aggregation is making oxygen transfer a limitation.
So we can sparge pure oxygen, which might increase.
So we can optimize the inlet gas phase concentration. What else? But that's an expensive strategy. You cannot afford to have pure oxygen inlet. So it is first the oxygen. So which means what? That oxygen transfer in plant cell fermentations are much more critical or the key limiting nutrient than the carbon. Generally for microbial fermentations, oxygen is not the limiting nutrient even in your Monod's model. That S is never O, oxygen. That S is carbon or nitrogen. At the max, when you work with products, you will find some other critical nutrient which might be affecting the product. But in plant cell fermentations or with hairy root cultures, I could find that because carbon was still available, it was not growing, which means that we need a model where now O2 becomes the limiting factor and not C. So that's how it shows that with plant cell fermentation, you cannot directly mimic what you know about microbial fermentations. So you need to first understand the culture itself, its requirement, and then work and use the right configuration. So impeller design or sparger design affects aeration and in turn oxygen transfer. So this is also critically needed. I'll not go into this, I'll just show you. So, but in, like for example, when we say that impeller design, high homogenization or power requirement, shear generation is needed, just saying doesn't help. Shear generation means what? How will you design an impeller taking into account shear generation, power requirement or circulation ability? How will you design? So generally what is done is you look in literature and get to the empirical correlations, which from where you first we need to define how would we define shear generation. How can shear be quantified in terms of these reactor operating parameters? So you can see literature, you will find there are many correlations, empirical correlations where people have correlated the shear forces generated with power input, with impeller speed, with impeller width. So one of the one which I showed you is one such empirical correlation which depends the amount of energy which would be dissipated onto the medium, depends on the power input, depends on the time duration and the volume of the reactor or the volume of the liquid. So eventually this power generation is also connected like in your power number. Given a power number, this power generation would eventually be correlated with the impeller speed, the diameter of the impeller. So now you know how these operating parameters will be impacting the shear force. Now first thing is you need to see the threshold of your culture. How would you see the threshold of your culture? You see, do a simple experiment. I'm just giving you an example. Do a simple experiment, keep on increasing the RPM. Check the viability of the cells. The RPM, once you know that this is the maximum RPM at which we can work backwards. At this RPM, what will be the shear generated? So which means this becomes your threshold shear force. So then you can fix up your RPM and similarly your power input or whatever, the volume of the reactor. If you need to know, suppose you want to quantify how much at this volume will be the shear force generated, or this may even help you to choose the right kind of impeller. Once you have fixed on that this can be the maximum shear which can be tolerated, then you choose that kind of an impeller or the power input such that you do not go beyond this shear force generated.